your answer sheet, and I'll have a couple extra credit questions for you. And then I think maybe the front page there, that would be 2 points each. And then you get some extra credit, that's 1 point each. And then this is how you're actually gonna start off. So before we start doing the answer sheet part, I'm gonna have you do a hands-on part. So this is what you're gonna have to do. Get your Petri plate out. I'll have a Petri plate for you, and I'll give each student their own little culture tube, and in there is gonna be a mixed culture. They're ready? Put your name on here. You want to write small around the outside. Then you're going to put the date on there, whatever. What's that date going to be? What's Thursday going to be? 18th. This is going to be the 18th. Wow. November 18th. Okay. Then you're going to use your Bunsen burner, right? And your sterile loop. A little flame on there, right? And you're going to transfer. You're going to do your street plate method. So get it on your plate, overlap, overlap. If you want, you can either do it this way, or you can leave it like that. Fine with me. As long as you spread them apart as best you can, right? First quadrant is when you add your bacteria. Then you flame the loop, right? To sterilize it and remove excess bacteria. After it cools, come back. Now you're going to make quadrant 2, and you need the overlap. Now you don't have to overlap the whole thing, just part of it. Then you're going to flame your loop again. Remove any excess bacteria. Let it cool. Then you're going to come back and do quadrant 3. And you got to overlap quadrant 2. Alrighty. Remember? When you're done, you know how you're going to put your incubator, put your Petri plate in the incubator upside down. So you're going to label the part of your plate that has the agar in it. And when you put it in the incubator, your label's going to be facing up. And the reason for that is actually really well demonstrated in this little number. Can you see all that water in there? So the reason we keep our cultures upside down is because a lot of times you'll get condensation and you don't want the water to drip onto the bacterial cultures on the agar. This way, if you get condensation, it stays in the lid. But then you have to handle it carefully so you don't get the water on the culture. I'll have the biohazard box out if I remember. And so you can drop your little culture tube into the biohazard box, because each student will have their own culture tube. So when you're done with it, just pitch it in the biohazard box, okay? Okie dokie. Then tidy up your spot and get yourself ready for the questions, okay? Like that. Now let's look at some slides that the questions might be. See here. So here's some different lab organisms I put on here to show you. So that wouldn't hurt because these are pretty common ones. So let's start with those and I'll go to my other file. Right? Do you know who that is? By just looking at them? That's an amoeba. Good. And these extensions are called?
Pseudopods.
Pseudopods. Good. And an amoeba is what kind of an organism? Animal, plant?
Eukaryote? No.
It's a eukaryote, but it's
animal.
protista. Protista. And it's protozoa because it feeds on other things. This is the shells of radiolarians. Radiolarians are amoebas. So imagine a creature similar to that lives inside and produces this shell. And then all these little holes, it can extend the pseudopods through the holes. And that way it can grab a piece of food or it can move. So radiolarians, you want to remember, those are also kingdom Protista. The only difference is that they make a shell. In this slide, we're using dark field microscopy because the background is dark. And that way the shells will glow and show up. This one, we're probably using phase contrast microscopy, but it actually looks like bright field because he's stained pink. Amoebas aren't naturally pink. So whenever you use a stain on your specimen, you're probably going to use bright field microscopy because how do you see the color of the stain? You got to have the lights on. Okay, I'll show you other pictures of Trichomonas vaginalis, but what's happening in this picture is they must have taken a swab and swabbed inside the vagina, and here are some squamous cells that sloughed off the lining, you know, the squamous epithelial lining of the vagina. And then all these little dudes are Trichomonas parasites. It's hard to see in this picture, but they're little single-celled organisms that are flagellated. And they're feeding on all that debris that's in the vagina, the dead cells and stuff like that. This guy you definitely should recognize, right? This is trypanosoma that causes sleeping sickness. One is more African, one is more South American. They're very, very similar. Same genus, Trypanosoma. So if you remember, Trypanosoma is a flagellated protista, just like Trichomonas. And this one, instead of living in the vagina, is swimming in the bloodstream because you see the red blood cells. Oh gosh, I just taught a class last night on the different blood cells, the different white blood cells. And remember when we did our trypanosoma slides, I took out those posters and they showed, I don't know if they're in the door anymore, they showed the different white blood cells. When you're looking at a blood smear, sometimes you'll see cells that stain purple. Those are the white blood cells. Okay, I think that's it for, oh no. Here's Giardia, another flagellated protozoan. This one actually has two nuclei, so some people think it looks like it has eyes, like a little face. So that's flagellated protozoan. This is ciliated, all around there's cilia, and this is paramecium. So there's protista with flagella, here's protista with cilia. Oh, here we go. Here's some white blood cells. And here's red blood cells. And plasmodium, actually those are not white blood cells. These are infected red blood cells, now that I read the heading. So in malaria, the malaria parasite enters your red blood cells. And as it bursts your red blood cells, that's when your fever spikes and your symptoms heighten. So malaria is genus Plasmodium. These are diatom shells, very similar to radiolarian shells, except that diatoms are photosynthetic inside. Oh dear, I think I feel a sneeze coming on. These are three-dimensional pictures. Remember what dinoflagellates are? They cause red tide. Here's euglena, which is green algae or phytoplankton, like plankton on SpongeBob. And he's flagellated and he's photosynthetic. That's a good one to recognize. That's definitely on your lab practical. Remember, Spirogyra is also protista, but it grows in a filamentous colony. And it's spirals of chloroplasts that are those little dots. Those are all chloroplasts spiraling through the cells. That's why they call it Spirogyra. When I was in college, there was a rock band called Spirogyra. They had some pretty music, if you Google it. Google oldies. This is a mold, magnified, growing off the surface of maybe a peach, something like that. This is the head of the fruiting body that's producing the spores. So when this gets knocked, all the little spores are going to fly off. This one is a yeast, Candida albicans. This is super magnified, so you can see the pseudohyphae. There is another yeast. This one is budding. See the new buds coming off? And this is Saccharomyces cerevisiae, which ferments grape juice to wine and mash to beer. Here's a mold growing on a petri plate. When you have a mold spore land on your plate or your bread, in this case they inoculated the plate by probably putting a inoculum of the mold here, here, and here. So all you need is one spore to land here. The spore germinates, grows hyphae, then the hyphae become complicated root system called mycelium. Once it's anchored into the agar and getting good nutrition, then it starts maturing from the most mature part of the colony out. And that's why the center of the colony is pigmented. Because this is more mature, it's making fruiting bodies and spores. And typically it's the spores that have the pigment. So it's the new growth that's white around the edges. But as this colony matures, this will become green and full of spores too. So in a bacterial colony or a mold colony, wherever you inoculated the center, that's the most mature part of the colony. Here's some fruiting bodies of a mold, and this one you can really see the head of the fruiting body has lots of little dotted spores on it. This is Aspergillus mold. I won't ask you the genus name. If you can look at this picture and tell me it's a mold instead of bacteria, I'm good with that, okay? And then if you could look at that and say, what do you think that is? Well, it looks like something magnified a lot, but those are the fruiting bodies with spores of a mold. This is actually the mold that causes ringworm. I'm like, really? Whatever. Don't worry about that one. Mushrooms. What's this? This one is a fluke. That's the schistosome fluke. Looks a lot, never mind. Fasciola and Clinorchus is very tiny. A lot of flukes are actually flat like this. And they have both sexes in the same worm.
What?
Yet, when you come to the schistosome fluke, it actually has male and female that have to mate, and it doesn't look like a steamroller went over it. It almost looks round to me. But to zoologists, when they examine them, their body structure and everything else, for some reason, they decided they're both flukes. But to me, it seems like they have some differences. So even though this is a fluke, just like that's a fluke, they look very different, don't they?
Yeah.
So the schistosome fluke is probably the most prevalent one worldwide, and so that's the one that we studied in our book. And tapeworm. Do you remember Taenia is tapeworms? This one is Dipylidium, which is actually a dog tapeworm. I think from eating rabbits and stuff like that. The scolex of the worm is the smallest part that attaches inside the intestine, and it has hooks and suckers for attaching inside the intestine, and then it grows proglottids out from the scolex, and they get bigger and fatter and more mature until the biggest, fattest proglottids are full of ripened eggs, and they will exit the anus and then perhaps be ingested by another animal, like another cow or something like that. Hopefully not a human, but I did have a little poster out for a while, you know, eat, never get fat, eat a tapeworm. So they actually tried back in the day to put a tapeworm scolex in a little capsule, and you swallow it and take it, and hopefully the tapeworm latches on, and then you live with a tapeworm in your digestive tract, and you'll always be thin. Yeah, a little gross, huh? That was on Urban Myth also. So, yeah, some people actually did it. There are some stories about a famous opera singer, and they thought that she had done that. I don't know. All right, these are roundworms now. This is human intestinal roundworms. This is doggy heartworm. That's a dog heart with the filarial worms in it, because they're long, thin, thread-like means filarial. So that's heartworm. That's a roundworm. This is a roundworm. Here's another roundworm that causes elephantiasis. So this is actually the microscopic stage of the roundworm in the bloodstream, I think. Or taken out of the bloodstream. You know your vectors, ticks, fleas. Got a lice.
No.
We got a mite. And this is the egg case of the louse. So this is a hare.
Oh yeah, those.
And the louse, this one is a female, I can tell because she has an innie. The male has like a pokey thing. All right, and this is the egg case that she attaches to your hair. Could even be a pubic hair, or a beard hair, or an eyelash.
Yeah.
Mosquitoes, probably the most important vector on the planet. All right, so you knew all those, right? That wasn't bad, was it? Let's see what else we got here. Let's try. See what this one looks like. Oh, I think I posted this.
Seriously.
Okay, let's see if we can zoom through these sections. See what kind of questions we would ask. Come on, light up. Oh, geez. That's a tiny one. Those are tapeworm proglottids in a pile of chicken poop. Even chickens can get tapeworms. What kind of world are we living in?
Yeah.
There's a bug for everybody, right? Alrighty. Tapeworm scolex. Okay, so this is a fluke. This is not the schistosome fluke. This is another kind of a fluke. And these are female parts. And these are male parts, all in the same worm. Well, actually, the male parts are kind of underneath. This is the head of a tapeworm called the Scolex. Scolex. It's got hooks and suckers for attachment. You have to go back to regular size in order to go to the next page. And here's a tapeworm, proglotid. Scolex is the tiny part. Proglotid actually gets much bigger than the scolex. This one's up inside the intestine. This is out down by the anus, ready to exit. Here's meat with cysts in it. So if you see your meat is full of cysts like this, but you're really hungry, cook it so that these get killed. Cook that meat thoroughly. This is a flat fluke, not the schistosome fluke, another kind. Here's a fluke, here's another fluke. This is a cross section of a worm, don't worry about that. This is the schistosome fluke. Yeah, which looks totally different from those flat flukes. And you got the male on the outside, wrapped around the female is actually on the inside. And guess what they're doing? The hokey pokey. I used the term hokey pokey because my dog was playing with her boyfriend this weekend. She's so in love with this dog. I'm like, you want to go visit Kobold? Can we get your leash on first? She pulls me all the way over to Kobold's house. I finally have to just let the leash go and let her run. And I was like, got to watch these two dogs now because she just finished her heat and I don't want to have puppies, so no hokey pokey. I had to keep an eye on her. They were sniffing around each other.
Oh.
And I was like, don't be doing that. Again, the schistosome fluke. Here's the male, here's the female. Oh, come on, let's get something interesting. Where's my next page? Ah! Here's the schistosome fluke life cycle. Okay? You don't need to know it for your lab practical, but you did on your test. What is the infectious stage? This little cercaria, that's what causes the disease. That's what enters your body. Here's the little cercaria under the microscope. Once it finds skin to penetrate, it will drop its swimming tail, and then just this part penetrates and enters the bloodstream. And here's a guy who has swimmer's itch, which means a cercaria of the wrong kind tried to enter a human. So this might be a cercaria of a fluke that infects a different kind of an animal. But the human happened to be there, and when it tried to enter the human, instead of it actually penetrating and causing full-blown schistosomiasis, his immune system dealt with it and made him itch and get these bumps to reject the thing, and we call it swimmer's itch. At some point, my dad had, the doctor called it sea lice. And I don't know what sea lice are, but maybe it's a similar thing. Because I guess they just have different nicknames for it, because they don't really know exactly what organism caused it, but they know basically what organism caused it. These are filarial roundworms. Oh no, these aren't filarial. These are puppy roundworms. Here's human roundworms.
Is that coming from the butthole?
Yep,
Oh.
poor kid. So obviously this is like worst case scenario. This kid's really infested with them. Here's the filarial worms, which means long, thin, thread-like. So this is what's going to cause heartworm and elephantiasis. These are roots of a plant, and the roots have been infested or infected with roundworms from the soil. I've seen a lot of this in Florida because when I first moved here, I worked for the Institute of Food and Agricultural Science, which was a division of the University of Florida, out there where Braden River High School is now. It used to be an agricultural research farm for the state, and I used to work for them. And what I would do is go out to various farms around the state in this beat up old car or truck And I'd take soil samples, and I'd also take root samples, and we'd examine them and try to figure out what kind of nematode worm in the soil was causing the damage to the crops. And then we'd have to make certain recommendations, how the farmer would fumigate and whatnot, because it's in the state's best interest to make sure that your farmers are doing well. And if farmers are having problems with soil nematodes destroying their crops, the state is going to help them figure out how to get rid of it so that they'll have a good crop yield, because then they'll pay more taxes and the state will be a lot happier and all that stuff. So these are damaged roots from nematode damage. Here's a tick, amoeba, radiolarian shells. Again, radiolarian shells, just not in such a fancy presentation, not so shiny. Paramecium, see the little cilia here? This one is the slide I'll probably show you. This is my favorite slide. This is a vaginal smear, an actual vaginal smear. So somebody has got her legs in the stirrups, doctor takes a swab, puts it in the preparation material, and they make a slide of it. And actually these are alive. So this is a dark field. I think somebody's rolling around in a chair on the roof.
I'd say.
Maybe. So here's some squamous cells sloughed off of the inside of the vagina walls. And these are the trichomonas parasites. See the flagella here?
I can't see it, Mother.
Can you
Yeah.
zoom in a little bit, please?
Oh yeah. Let's see. if we can-
Zoom that sucker and there you go.
Can you see the flagella here?
Oh, okay,
Oh, okay.
okay,
And
okay.
here's
It's-
flagella here.
It's not as highlighted.
And here's flagella here. And why are they glowing? Because this isn't just dark field, it's actually phase contrast. So when these guys move,
It's been stained.
because they're swimming and eating, that moves the water in the wet mount and it makes it kind of glow around where they're moving. And that's how I know they're alive.
So,
They're magic.
so it's trichomonas.
So this is a vaginal smear of Trichomonas vaginalis. All these with the glow around them are the parasites, and they're munching on these dead, sloughed off squamous cells in the vagina. Lovely.
How beautiful.
Come on. I want to go to the next slide. Diatom shells again. Diatoms are photosynthetic, radiolarians are not. Euglena, E-U-G-L-E-N-A, Euglena.
I need to know how to spell this.
Euglena, there's a huge group of euglenoids. The genus I just call Euglena. They're flagellated
But she's recording it.
and they're photosynthetic. So they are actually able to chase down food and eat it like a heterotroph, but they can also do photosynthesis like an autotroph. So Euglena's really a popular organism because he's multi-talented. Yeah?
Oh, I was thinking it was photosynthetic because it's green.
Yeah, green. Green is what makes it photosynthetic, absolutely. And here's some more photosynthetic critters, they're green. This one is Spirogyra, which is actually a chain of cells of Protista. And this one is Oscillatoria, which is actually stacks of cells of bacteria. My point is, they occur together in the pond. They're all just green algae, green pond scum. I love to watch the pond at Payne Park, where all the hobos sleep, you know.
No comment.
And they're sleeping on all the duck poo and everything, because there's a lot of bird shit around there.
And dog.
But I love to watch that pond, because at a certain season, all of the azole plants, they're water plants, they're covering the entire surface of the pond in like a yellow-green. And then if you go back like 2 weeks later, they've all died down and dried up and gone away. And other times the pond is covered in duckweed. So it's like you see the seasons in the pond
Wow.
with whatever kinds of algae are growing there. I don't know how many people pay attention to that kind of nonsense, but I do. because I guess I just always was curious about nature. I always was interested. Here's more green algae pond scum. This one is spirulina, and this one is oscillatoria. They're both cyanobacteria. So some protista can be photosynthetic, and some bacteria can be photosynthetic, and that's what makes up pond scum. Photosynthetic bacteria, protista, and even some little plants like duckweed. More spirogyra, which is photosynthetic bacteria, cyanobacteria. And this would be probably a wet mount. Can you tell that it's kind of in a liquidy background? And here too, it's kind of in a liquidy background. This is a whole bunch of oscillatoria. Because when we look at it in our microscope, we're looking at this filament of cells. Yeah, but if you look at the big picture, they're all just together. And when you look at the whole pond, it's just got a whole bunch of green stuff in it because it's all clumped together. Yeast. This one is Saccharomyces cerevisiae, which is baker's yeast or brewer's yeast. You can buy it at Publix if you want to make bread or something. Me, I just go to the Publix bakery and I let them do all the cooking and cleaning up. Because if you're going to cook something, you're going to have to clean up afterwards. And so I rather let Publix do it. I'll pay the extra money not to have to clean up my kitchen. And Publix does a pretty good job on most things. Did you ever have the Publix carrot cake? That's really good.
Oh, wow.
Yeah, that's good.
Carrot cake.
Carrot cake.
On Easter.
On Easter, that's the only time you get Publix carrot cake? That's a good idea for Easter, I have to say, but I've given it for birthday cakes. What? But some people don't like carrot cake, but most people like it. Anyways, these are budding yeast cells. Easy peasy. Got anything more interesting? Ah, now we're getting interesting. Here's a Petri plate. What technique did they use?
Street.
Street plate. And how many quadrants do they have?
Looks like
Three.
two. Looks
I think there's only two.
Two.
Here's the first one, and then here's the second one. There's the overlap, and then this is where the overlap stops. They didn't quite overlap up into there. It's okay. And then they still just zigzagged, I think. And they did get some isolated colonies, I would say. But maybe if they had done a third quadrant, they would have had more isolated colonies. For our purposes, this plate will be just fine.
Okay.
And how
Okay,
many
okay.
different kinds of bacteria are growing here?
One.
I
No,
would
two.
say one, because it all looks the same to me.
Yeah.
So I would say this is probably a pure culture.
Pure culture.
And again, in the lab and stuff like that, sometimes answers can be kind of subjective, you know. You might see one thing, you might see something else. This is really super magnified.
What is that?
I want you to see, this is the tip of somebody's penis.
A head?
And here is the yeast infection. See it?
Yeah.
So he's got a little yeast infection, and it's like under the fold of skin. But let's see what else is going on with this gentleman. What the heck
Is
are
that a
these?
HPV? I knew it.
I think he's got one here. I think he might be getting a few here. I think this dude's got a little HPV going on. And what the heck is that on his finger?
Oh my lord.
Is that a,
I think it's a wart too.
but what do you,
I think they're all warts. I think he's a warty guy. Now he's got yeast too. But overall, he looks generally healthy. We just got a few issues to take care of. The HPV, they can remove those warts, but many times they'll grow back. Here's our yeast infection in a female, just to be fair. In the science department, we were having a conversation about how everything changes, including scientific terminology. There is a big push right now to standardize scientific terminology. It's about time. Can we standardize copy machines too, please? All right. So, in the olden days, Dr. Fallopian discovered the fallopian tubes. So they named them fallopian tubes. And they taught all of us that they're called fallopian tubes. The younger generation comes up and says, why are they called fallopian tubes? Oh, well, because they were discovered by Dr. Fallopian in the 2000 BC or whenever, a million years ago. And they were like, I can't remember that because it doesn't make sense. We're going to call them uterine tubes.
Oh,
Yeah, right.
okay.
We're learning it now.
Fallopian to uterine to
Yeah, that's all it is.
It's all about the ego.
Always evil.
It's all about the ego. In the olden days, you discovered something, you had to have your name. I even remember my own professor in graduate school. They wanted to have their name on an article in Nature magazine or whatever, They had a very big ego. And that's why everything's named after everybody instead of having a name that makes sense, that people can learn and remember because it makes sense. This guy has cutaneous, which means on the skin, candidiasis, which means an infection of Candida albicans, which is normal flora. He's just got an overgrowth. Here you don't see the white pasty yeast like you did in the vaginal or the penile yeast infection. Here you just see the irritated red raw skin. And why is the skin red raw and irritated? Because those pseudohyphae have started penetrating. So a lot of times it'll look like a red rash. You're sponge bathing your old patient, you lift up that floppy boobie, and you're like, what's going on under there? And it's all red and prickly, probably a yeast infection. Sometimes you don't see the white yeast, you just see the red irritated skin. But then again, sometimes you will see the yeast, particularly in oral thrush. Oral thrush can occur in babies a lot because their immune system is not fully developed. And when a baby sucks on a bottle or on the mother and they leave a little milk or juice in their mouth, that gives the yeast a chance to flourish and their immune system's not quite ready to keep them in check. So a lot of times babies will end up with oral thrush. Sometimes I had a woman in my class and she actually got like a mastitis from it. And this happens with animals a lot too. Mastitis is an inflammation of the boob and the nipple. So because the baby had oral thrush, she also got thrush on and around her nipple. And it was apparently very painful. And so they had topical medicine for inside the mouth and on the boobie, and I think she also took an oral antifungal. And then don't forget your immunocompromised patients, like AIDS patients, patients on chemotherapy, that can throw their balance of flora out of whack and they end up with a yeast infection. And yeast infections, you'll probably see a lot of in healthcare. The bummer is, because your patients are often weak, the yeast infection can actually be very hard on their system, and sometimes the yeast infection can become systemic and kill your patient. And there is a yeast superbug. I'm not sure I'm going to spell this right, but you can probably Google it for the right spelling. Candida auris, I think it is.
AU.
So it's golden?
No, is it AURIS, auris or something? It's a species of Candida that is highly resistant to the antifungals, which is a bummer. What, I spelled it right?
Yeah.
Good for me, yay, that's one thing the Prevagen's starting to kick in. What's up, Julio?
No, I was saying hooray.
Oh, good. And here's another picture of Candida. Like, it's got all the little budding yeast parts, but it's also got the pseudohyphae showing. By the way, this would be like a dark field or a phase contrast micrograph. I will ask you questions about that. So if you see a slide and the specimen has color and the background is bright, the light is on your microscope, that's probably bright field microscopy. When you see the background is dark, it's probably dark field microscopy, And a lot of times, it can also be phase contrast microscopy, which is a little more grayish than black. And in phase contrast, you tend to get a little bit more of the glowy in the background. But if either answer would be acceptable to me. So if you say dark background, dark field, I'm good with that. Close enough. You know the difference. So I just put this up. It's something I stole from the internet because it shows this one. These are squamous cells. And this is the Candida albicans. And here's the budding yeast, and here's the pseudohyphae. So I put this in just because this has another nice picture here. Disease risk factors: AIDS, diabetes, surgery, catheters, antibiotics, oh, let's not forget about that one, neutropenia, burns, and dialysis. Part of normal human flora, they're opportunists of the oral cavity, GI, which is gastrointestinal, and genital tract. Here are several different species. Candida albicans is the one I usually talk about. Yeasts have pseudohyphae, elongated single cells with constricted ends, and true hyphae, septasia, whatever. And then this one, you probably can't see it, but that looks like Staphylococcus that's stained purple to me. And I don't know what that is, but anyway, if I had that one there. Here's a mold on a peach. Can you see the surface of the peach?
There.
You know how the peach has that little indent where the stem is?
Those are the fruit
Right, so
bodies.
this is a peach, and this is Rhizopus mold, which is a black mold, and that's because the spores are black. So here's the mycelium growing into the peach, and then the fruiting bodies coming off the surface of the peach, producing these spores in these round ball-shaped caps. Fuzzy peach, fuzzy navel. Oh, that's what it is. It's a fuzzy navel, because I guess is that the navel of the peach?
Yeah.
Where the pit is? I haven't had one of those in years.
I'm not sure.
Get the eye.
All right, mushrooms. It's like a big mold. The mushroom is the fruiting body. Here's the hyphae making up the mycelium, anchoring it to the ground. The fruiting body grows up and produces spores. So you know how mushrooms work. Molds work the same way. This is not per se on your lab practical, but I found this on the internet, and I thought that was really cool because it's got everybody all on the same page. It's a good little refresher for the different bacteria we study. Okay, this one you'll see on your lab practical, so jot this one down. What's going on here? This is a vibrio-shaped cell. Vibrios are a curved rod like a hot dog or a banana, and they have a flagellum. And one of the most notorious vibrios
Unreal.
is Vibrio cholerae, which causes cholera. Now remember, Dr. John Snow made a map of all the cholera deaths in London, and he found that they all centered around a contaminated pump on Broad Street. He had never even heard of bacteria. This was before they discovered bacteria. All he knew was that was the source of the epidemic. And that's what we're going to do in our party next week. We're going to have a party, and at the party, there's going to be an epidemic happen. At the end of the party, we're going to have to figure out the source of the epidemic. Who came to the party with a bad germ and spread it to everybody else? Yep. Yep. Give him little devil horns to go with his
I got some.
scrubs. Actually, I've already thought of my Halloween costume for next year.
What?
I always make my Halloween costume out of like my yoga clothes.
Wow.
So this year I wore all black yoga clothes, but next year I'm gonna wear all red yoga clothes. And I'm gonna be Little Red Riding Hood, and my dog's gonna be the big bad wolf.
Oh
Dressed
yeah.
as grandma.
He's brown.
He's gonna be super big by then.
Yeah. They keep telling me that she's gonna grow like 15 more pounds at least. I'm like, are you kidding me? She's already 70 pounds. And then I can also use the red to be a little devil.
You'd be the devil's
Yeah.
advocate.
And then I can make her into a flying monkey or something like that. What's happening here is it's an artist drawing. They're trying to show you this is the lining of your bowels, and they're swimming along the lining of your bowels. That's where cholera causes infection in your bowels, and it causes a severe dehydrating diarrhea, which is what will kill you. And some cholera produce toxins too and stuff. So depending on the strain you get, could be very, very serious. And some people will actually carry some strains of cholera and can even infect other people with their bad hygiene. But their body's kind of dealing with it and they harbor it. So it's like a typhoid Mary, only it would be a cholera Mary. Yeah, isn't that scary? So there actually was a case of that where this church was sponsoring people from another country to come and live here for a while with a family from the church. Well, this woman came and lived with a family, and little by little, everybody in the family started getting sick, and they all got cholera. Turns out the woman was a cholera carrier, and she was helping them around the house with the chores and the cooking and everything, and she didn't practice good hygiene, and she infected the whole family. So, but at least then they found out that she was a cholera carrier, so they could heal her also. So here's another Petri plate. You'll get stuff like this on your practical. Again, I see a streak plate method. It's a little hard to tell what's going on. They missed a whole area they could have used, but they did get isolation. And look at these weird shaped colonies. Like here's the center, and then it kind of bunches out. But it does look like a pure culture to me. I think it looks like they're all the same. So I'll ask you questions like which one is a pure culture, which one is a mixed culture. Now we're getting into some nitty gritty, so I hope your brain is warmed up. All right, these are bacillus-shaped cells, right? And the endospores are green. So here's a cell with an endospore inside. Here's an endospore after the cell has died and disintegrated. And here's a cell before it's even made an endospore. This is different. This is a capsule stain. Notice that the cell is stained, the background is stained, and the outer gooey capsule stays clear. Function of the endospore, dormant stage for soil bacteria like genus Bacillus and genus Clostridium. Function of the capsule, stick to host tissues like in pneumonia causing diseases. This one is Klebsiella pneumoniae. Other bacteria known for having a capsule, Streptococcus. The capsule is clear, the cell gets stained, and the background also gets stained. Because the background gets stained, it's considered a type of a negative stain. So a capsule stain is a type of a negative stain. What's the other functions of the capsule besides sticking to host tissue?
Hide.
Hide from our phagocytes,
Yeah.
avoid phagocytosis, and also can stick together with other cells and form a biofilm. Right. Here's another picture of Klebsiella pneumoniae with capsules. It's just the color of the dye is a little different. The capsule is the clear area. The background is stained and the cell inside is stained. Does this help refresh your memory?
Would that be in bright field then, because we can see the color?
Absolutely, good job. This is an artist drawing, but I like it because it's real clear that this is a capsule. A lot of times students make a common mistake and think that this is an endospore inside a cell. And I get that. I get it. It is hard to kind of tell, but I want you to try to determine the difference when you look at these pictures. Okay, what do we got going on here? Shapes and arrangements of bacteria. So here are tiny single bacilli. You might even call them coccobacilli if they're roundish like that one. but I would say they're tiny single bacilli. Whereas here we have large bacilli in a chain. So this would be a streptobacillus. So sometimes you'll get a question I'm looking for, what is the shape and arrangement, which is the cellular morphology. The cellular morphology here, you could say a chain of bacilli, or you could say a streptobacillus. Here you would say single bacilli. And what kind of a slide is this? This is definitely a negative stain.
Oh,
Because it's like nigrosin in the background and the cells are white, so this is definitely a negative stain. Or it could be a dark field microscopy, but sometimes that's a little hard with bacteria. The stain actually works better. Dark field microscopy tends to work better if you have like a wet mount of something alive, maybe even. But, you know, there's no rules. However, your microscope helps you see what you want to see. So on my microscope, I try everything. I'm like, I saw it in bright field, let's try dark field, phase contrast, let's try this kind of a stain, let's try that kind of a stain. Here's another negative stain, and you have a streptococcus here, okay? Cellular morphology, streptococcus. Here's your AFB slide. Remember when we did these? We're checking for acid-fast bacilli in our patient's sputum. Sputum, S-P-U-T-U-M, sputum. When I was a little kid, Russia launched a satellite called Sputnik. And a lot of people call it sputum. But it's sputum, like mucus. What's in the positive control of your AFB slide? Something that looks like tuberculosis, Mycobacterium. So the positive control is Mycobacterium, because we're checking for Mycobacterium, which is acid-fast bacilli, in your patient's sputum. And in the negative control, you could actually have anything that's not Mycobacterium. But the one they put in there is E. coli, just because it's about the same size and shape as the Mycobacterium. We just like E. coli. And speaking of E. coli, here's E. coli, you know, because on EMB agar, eosin methylene blue agar, E. coli metabolizes it when it takes up the lactose, it takes up the methylene blue, and that metabolizes to shiny green. It's a unique reaction that only E. coli and strong lactose fermenters do on this plate. So when we use EMB agar, in fact, I had a friend when I was much younger, and he had a job, he would live at college, and then in the summer we'd all come home for vacation. And then the next semester go back to college. Well, his summer job at home was to check this little reservoir we had in our town. He would literally dip his sterile swab in the reservoir and swab these plates and stick them in the incubator every day. And then he'd check the plates from the day before. The next day he went to work. And if he saw any colonies of shiny green, they knew there was poop contaminating the reservoir, and they had to shock it with extra chlorine. So they would pump extra chlorine to the reservoir. And sometimes this would happen after a heavy rain. Because when there's a heavy rain, all the cow shit on the pasture, all the dog shit on the street, all the homeless people, stray animal poop, all the dirt and the litter, and everything on the ground gets washed in to the reservoir and to our wells and into the bay and into the Gulf and all of that stuff. So that's why it's important that we keep our streets clean, right? But it's hard when you have no control over people pooping in the street. I mean, it's just one thing that just, I can't even stand it when I see somebody spit a piece of gum on the street. I mean, that just irritates me no end. I'm like, how dare you spit gum on the street? Some poor little squirrel is going to come along and get his foot stuck in there. Don't you hate that? You got a brand new pair of shoes and you step in somebody's damn chewing gum. Doesn't that piss you off? Or it gets on your brand new car and then you're trying to get it off the... So, anyways, it's E. coli. But the bigger question, What family does E. coli belong to?
Enterobacteriaceae.
Thank you, thank you, thank you. Well learned.
I can spell it though.
It's a big one. If you got the entero part, you're good.
Oh, okay.
So just think enterobacteria aceae.
Okay.
A-C-E-A-E. It's like a Latin ending. And that's the other thing. A lot of our medical terminology, this one doesn't want to get bigger. A lot of our medical terminology actually
Latin.
comes from Greek and Latin.
Yeah.
Here's another street plate. One,
Ooh,
two,
two,
three, four
four.
quadrants.
Cinco.
It's 4?
Got a little bit of isolated colonies in here. Looks like a pure culture. This one is the EMB agar plate for comparison. Right here's your E. coli. This might be another lactose fermenter, just not as strong as E. coli. And then this one, it's growing there. It's hard to tell, but there is growth, but it's not using the lactose. So it's probably not even growing that well if it can't use the main ingredient, you know. If all they're serving for dinner is meat and you're a vegetarian, you're going to go hungry. I've been in that position once. Sometimes you look back on your life and it's like, why did I ever subject myself to that? Because you didn't know going in that it was going to be that bad. But that's what happens when you try to cooperate with other people. This one is the McConkey agar. And when you get the milky pink reaction, that means it's also using lactose. And here is your Salmonella Shigella agar. All three of those have lactose in it. If you have a lactose fermenter like E. coli, it'll turn milky pink, like the McConkey agar did. But if it's not a lactose fermenter, you won't get the milky pink. Instead, you'll get growth, but it won't be milky pink. And if you get growth and it's black, then you know it's salmonella. Yeast pathogenic, yeast pathogenic. We test them on Salmonella Shigella agar because we want to know which pathogen is it. If we get E. coli results, well, that's not really a pathogen. Maybe we can find a solution to this problem. It's just E. coli. But don't forget, E. coli can also cause disease. Here's your Salmonella Shigella agar. The black coloration comes from Salmonella's ability to produce hydrogen sulfide, which smells nasty. In fact, I have a pond behind my house, and it's the sulfur water. When I first moved in, I was like, oh God, do I have to smell sulfur water all day long? Disgusting. But you know, now I don't even smell it anymore. I'm so used to it. Yeah, it's funny the things you get used to. I used to think it was horrible. Now I don't even notice it. This is the mannitol salt agar. Mannitol salt agar is done for checking for pathogenic staphylococcus. So on this plate, you want to grow staphylococcus. And if they come out turning the agar yellow, they're pathogenic staphylococcus, like Staphylococcus aureus. If they grow but they don't turn the agar any color, it stays pink, then they're non-pathogenic staphylococcus, like Staphylococcus epidermidis on our skin. So here is another example of a mannitol salt agar plate. Here's your pathogenic staphylococcus. Here's your non-pathogenic staphylococcus. And here's your E. coli. Couldn't even grow. So mannitol salt agar has salt in it, and it's the salt that the staphylococcus like that the E. coli hates. So the selective ingredient is the salt. The differential ingredient is the mannitol sugar. If it can ferment the mannitol sugar, it will excrete acid waste and turn the agar yellow. If it can't ferment the mannitol sugar, the agar gets no color change.
So Staphylococcus aureus is the one that fed the ham? The salt.
Yeah, that affects ham. Yep, it can tolerate the salt even of a ham.
No.
Okay, yellow, right? So what's your biggest clue right here? Color,
Yes.
yellow pigment. It's Micrococcus luteus. Luteus means yellow.
I don't even know the difference between
Okay,
gold
this
and
is a streak
Orange
plate,
is good.
but
Yellow
the
is
streak
good,
plate
yes.
but like,
doesn't
I
look
can't
like it was
tell
done
which
very
is
well.
gold and yellow.
They put all their bacteria, here's quadrant 1, then they got this, and then they got this. They just started with way too much bacteria on their loop, that's all. This plate's got way too much bacteria on it for good isolation. But they got lucky, they got a couple of isolated colonies there. So I just would say here, just don't put so much bacteria on it first. Here's your Micrococcus luteus, right? This one is cellular morphology. It's a staphylococcus. In fact, it's a tetrad. Let's go back to the colonial morphology. Colonial morphology, circular, shiny, smooth, yellow colonies. Colonial morphology. Take a little sample, stain it purple, put it on my microscope. Cellular morphology. I have staphylococci, little tetrads of cocci. Here's some more cellular morphology. These are staphylococci. Here's a little tetrad, nice one right there. Here's the difference between luteus and aureus.
Here you go.
I knew I had this picture in here somewhere, I just couldn't remember where. I've been dying to show you all semester. Yeah, luteus is yellow.
Mustard.
Okay, yellow.
Aureus is golden.
Honey mustard.
Isn't that funny?
I thought it
So it's darker. Gold is darker. I want the yellow.
And look at their streak plate method. That's the first quadrant, then they do the second, the third, and the fourth
was- That's neat.
into the middle. So they kind of go around the outer edges of the plate, and then the fourth quadrant, they do their overlap and then go into the middle. So that's an interesting technique too. Yay! What bacterium has red pigment? Serratia, good.
Shit.
So what's your biggest clue? Red pigment.
No. Serratia. Serratia has red
There is Petri plate art. If you Google it, they use different augers that make the bacteria turn different colors, and then they use different species, and they actually paint pictures in the Petri plate with the bacteria.
Wow.
It's called Petri plate art. We'll have to Google someone again. Serratia again, pure culture. This one is actually, if we look really close, I think it's either a mixed culture or a contaminated culture. Because see this little yellow one here?
Yeah.
And then the white ones? So I think that's a contaminant. Come on, where's my thing? Is that the end?
No.
Here's a mixed culture, yellow and red. Right? Could you make a pure culture if you sampled just this one, made a new plate? That'd be a pure culture of the yellow ones. If you sample just this one, make a new plate, that'll be a culture of just the red ones. So you can separate these two and make two separate pure cultures from this plate, which has the mixed culture. And that's the reason why I did the streak plate method, so we could get them to separate from one another into individual colonies. And another serraceous streak plate. And what the heck is this thing called?
I forgot.
This was your
20
API test strip.
API, 20.
And the test we did was 20E,
20E.
which means 20 tests for Enterobacteriaceae.
API-20E.
It's your API test strip. Here it is again. Here is what? What's this one called?
Antibiotic.
Kirby-Bauer disc diffusion antibiotic sensitivity test. Kirby-Bauer method. Everybody knows Kirby-Bauer method. You
Kirby.
put your antibiotic discs on your bacterial lawn. measure the size of the zone of inhibition all the way across, compare that diameter to a chart for standardization, and then it will determine if this size of zone of inhibition indicates that this particular species of bacteria is sensitive to this antibiotic or not. So the bacteria are resistant to A, they're resistant to C. It's not a very complete inhibition around F, so I wouldn't even measure that one. D, it's a very small zone of inhibition. B, it's bigger. G, it's bigger. And E is the biggest. But we still have to compare our diameter measurements to that standardization chart before we absolutely can pick which antibiotic is the best for this particular bacterial infection. Also, we're going to have to consider patient factors. What if we choose B? Can the patient take B? Are they going to be able to tolerate B? So sometimes you might have to choose a different antibiotic that maybe is not as effective against the bacteria because that's all your patient can tolerate. And in that case, they might give her two different things together to help her out. Two things that she can tolerate that will have a synergistic effect. All right, here's another Kirby-Bauer disc diffusion plate. That's a big one. Got a lot of discs on it. Ah, the serial dilution. Don't worry about this. I'm not going to make you do any math on your lab practical. Just remember what a serial dilution is. It's like a serial killer.
Door to door.
One after another. I'm diluting it down, diluting it down, diluting it down. And as I dilute it down, I get less and less growth. Well, I want to pick my plate with between 30 and 300 colonies on it, count up the number of colonies, and use that in the titer formula to calculate the concentration of my original culture. I want to know the density of bacteria per milliliter in my original culture. Oh, what kind of plate is this one?
Whole plate.
It's a pour plate. Good job. And you know this because you're seeing little colonies as well as large colonies. This has to be a facultative anaerobe in order for both of them to be able to live. Can live with air, can live without air, just not as well, right? If it were an anaerobe and we made a pour plate, we'd get all colonies underneath the agar, because the ones that landed on the surface would be killed by the air, so only the ones underneath the agar would be able to grow. If I had a strict aerobe like Pseudomonas, they'd all grow only on the surface. The ones that got trapped underneath would be killed off. So when you do the pour plate method, it's important that you consider whether the bacteria you're using in your pour plate method are facultative or anaerobic like Pseudomonas, or anaerobic like Clostridium, or aerobic like Pseudomonas, right? Because it's going to make your plate come out different. But I like to use E. coli on these plates because you can see the difference in their metabolism with oxygen and without oxygen. Makes a big difference in size. But they're still alive. And if they can get to the point where, let's say I cut them out of the agar with my scalpel and I maybe spread them on top of a plate where they'll go back and use the air and they'll grow to bigger colonies. Does anybody recognize what kind of plate this is? You're swirling, spreading. So you squirt your solution with your bacteria onto the surface, and then you spread it with your spreader. So this is a spread plate. All the colonies are on the surface. They're about the same size, and you can almost see the circular spreading pattern that this person did. Here's another spread plate. Here's a streak plate, kind of a unique method, but very neat, isn't it? Some people just have all kinds of talent and unbelievable. Here's another serial dilution, only this one is for a virus or a phage, right? It's the same thing. Dilute it down, then you're going to plate it out, And I think with the virus, you just used a spread plate. No, you did a, what did we do? We did a pour plate, didn't we?
Yeah.
Here's the phage, the T4 bacterial phage. He's up close, these are in the background. So this is actually the cell he's landing on. This big green one at the bottom. There's the head or the capsid. This is called the collar, tail, and tail fibers. That's the T4 bacterial phage that infects E. coli. And here is your phage plaque assay plate. There's the cloudy agar, a bacterial lawn. The agar is full of bacteria. Here are the phage plaques where they've replicated inside of and lysed the E. coli. Count up the number of plaques. Use that as N in your titer formula. Then you calculate your titer in phages per milliliter. Alrighty, this is a summary of the lytic cycle of a virulent phage because it's killing the host cell. Oh, here's another one of those summaries I downloaded from the internet of all the different human viruses. This is bullet shaped, right? So that's the rabies virus with the bullet shape. Here's different shapes of viruses. Ebola virus, can you see that gray one with the red on it and that blue one with the red on it? Those are our Ebola virus. Bacterial phage. The other viruses, protein spikes on these. Oh, interesting. Coronavirus is called coronavirus because it has protein spikes coming out all around it, so it looks like a crown. So they called it coronavirus. Whoop. Get back to another picture. This is the lysogenic cycle. This is the lambda phage. It has a tail, but no tail fibers. But it still infects E. coli, but it can go through a lysogenic cycle, producing lysogens that contain a prophage. And lysogenic conversion is when the prophage genes change the bacteria to having a new characteristic, like the ability to produce a toxin. Here are simple squamous epithelial cells from the inside of my cheek. These are cheek cells, nucleus, squamous cell, and look at my oral flora all stuck to my cheek cells. So I want you to recognize your cell and your normal flora, your normal bacteria. A lot of time they like to stick to the old cells because they're going to break them down. This guy has flagella, right? He's a spirillum, not a spirochete. Spirochetes have an axial filament on the inside. Spirillums have external flagella. The flagella are for swimming, movement. Here's that same slide again. Here's more flagella. Who's flagellated? A lot of bacteria, but this particular extra large one is Spirillum volutans. We can see his flagella really well because he's so big. E. coli is covered in flagella, but E. coli is so small, you'd need an electron microscope to be able to see his flagella. Alrighty, more squamous cells. Recognize your squamous epithelial cells with their nucleus. These could be contents inside the cell, but sometimes you'll see little dots too that are your normal flora just stuck to your cells. I want you to be able to recognize your cells like you'd see in anatomy with bacteria and fungi and stuff that might be mixed in with your cells. This one I actually have posted in the lecture as extras to go with part 3. I just found this somewhere and I liked it because it shows you a eukaryotic cell with a nucleus with a nuclear membrane. Inside the nucleus is the double helix DNA, which will open up for the length of one gene and make a single-stranded copy of RNA in transcription. Once the mRNA copy of the gene is made, the gene closes back up, stays safe in the nucleus. But the mRNA exits through a nuclear pore, and so do the 2 other kinds of RNA, which are also being made in the nucleus, meanwhile. And this goes out to the cytoplasm, where it weaves through a ribosome, and ribosomes contain RNA and protein. And as the mRNA weaves through the rRNA, the tRNA, or transfer RNA molecules, come in, do complementary base pairing with the codons, with their anticodon, and carry the appropriate amino acid into place and add it to the growing chain of amino acids. Until what the gene told the cell to make, this chain of amino acids, is complete, then the mRNA, this whole thing disassembles, this floats free in the cytoplasm, finishes twisting, bending, folding, assuming its shape, and once its shape is complete, it's a functional protein or enzyme. So, this is essentially the central dogma of genetics. DNA is transcribed to RNA, which is translated to protein. And that's how we think life goes on. Trypanosoma again, right? He's flagellated, he's got an undulating membrane, swimming among your red blood cells in your bloodstream. Come on. Why aren't you changing? Are you my last slide? Where's my arrow? Yep, that's it. We're done. Perfect timing. Okay, so review that stuff.
I'm scared.
Don't be scared.
Are all the questions gonna be